say clear if you are a lesbian?
Because why not?
All right, so moving on with biochemistry. I wanted to go over this again because it was kind of rushed at the end of last lecture, and it's a good review anyway. So we want to talk about how ATP synthase works. I want to talk about the 3 steps essentially. All right, so what we're going to do is we start out. I've got all 3 steps up here. There's just a kind of goes back to where it was is the one that I've got below the screen there. I hope you guys can see all that. And so we can think about steps 1, 2, 3 here. And what we can do is focus on the purple subunit here. So we've got step 1, step 2, step 3. And remember that each of these subunits here, they're going to go through different conformations. Each of those is, or I should say domains, okay? We've got a purple domain, a light blue domain, and a light green domain, okay? And in terms of those domains, the alpha and beta are subunits, okay? We're talking about a subunit, we're talking about a peptide, an individual peptide chain. So if we think about what's going on with 1 here for purple, okay, so we'll focus on purple. They're offset if you look at what their affinities are here. So this right here has a low affinity for ATP, so it's not shown here, but it has released ATP, like that, okay? So I'll put a 1 down below once we get there, but it's a low affinity for ATP. This one has a high affinity, that shape has the high affinity for ADP and phosphate. And then this shape has a high affinity for ATP in such a way that it smashes the 2 together, okay, to make it ATP. And notice that at any given point, one of these is in one of the 3 conformations, all right, and they're just going to all shift through. They're not, they are working slightly out of phase from one another. So 1, the domain has a low affinity for ATP. All right, and that just happened for this top one. We want to do 1 again. 1 is down here, you see, right there. So put another 1 here. And I don't care what order you put these in as long as they progress through the correct order. I don't care which one you start with, as long as they progress through the correct order to make us some ATP. So 2, okay, actually let's do a 1.5. Maybe it's not going to let me do 1.5. So what happens then in between these steps is 3 ATP pass through the membrane. I'm going to say the C subunit wheel, turning it, and which turning the gamma subunit, okay, and that causes our domains to change shape, causing all the domains. Lorenzo. Did I say, what did I say?
Last time.
3 ATP. Apparently it's stuck in my brain wrong. Thank you. 3H plus passed through the C subunit wheel. Okay, we're raking ATP here. I'm going to copy this so I don't do it wrong again, because it's going to happen again, right? So then we get to 2. All right, at 2, the domain has a high affinity for ADP and phosphate, and it binds them. Okay, each time, we'll go ahead, we'll just paste in this thing. So now we have another 3 protons that are going to pass through the membrane using those C subunits, that wheel, right? And that's going to turn our gamma subunit, which is going to cause all three domains to change shape again. And focusing on the purple domain, the domain has a high affinity for ATP such that I'm just going to say it joins ADP and phosphate together. Okay. Then, once again, we have 3 protons are going to pass through the membrane, through the C subunits. They turn everything. All our subunits change shape, and then we're back to 1. And it repeats over and over again. So we're back to 1 where we have a low affinity for ATP, and that ATP that was just made can now be released, getting us ready for another cycle. Does that make sense how that works? Okay, you're responsible for being able to describe that. Yes?
So the right series says it has a high affinity for ADP and PI.
Yes, and phosphate.
Sorry, phosphate. And then it binds to the ATP. But then in the other tree says it has a high affinity for ATP.
Yes, so it's essentially, it has a great shape for binding ATP instead of ADP and phosphate, and it causes ATP to be made. You don't like that?
No.
Okay. Yes?
Why are we focusing on like the purple? What's the difference between the purple, the blue, and the green?
That's a great question. So what's the difference between the purple and the other colors? There is no difference except they're just offset from each other. But if we focus on the blue, we change what number was what, right? We'd start with, what have we got here? Tight binding to make ATP was number 3, right? So if we were doing blue, this would be 3. This would be 1, right? It's being released. This would be 2. So we're just offset. So 3 protons going through the membrane causes 1 ATP to be made and released, right? One ATP. So, but we've got all 3 subunits going at the same time, right? So if we go here to here, this is 3 protons, 1 ATP is made and it gets released. 3 more protons, 1 ATP is made and it's released.
So it's technically it's like per single domain it's like 9, but overall it's
For single domain it's 3.
well because
but you have 3 of them moving. We may be talking semantics here.
okay.
You're going to get 3 ATP per 9, right? Right? So, okay. Other questions or thoughts on that? Are people confused on it?
Wait, can you explain that one time? 3 ATP per 9.
So it's going to be 3 ATP per 9 protons crossing the membrane because we'll have gone a full cycle for one of these, but for each time you change the conformations, you're changing the conformation of one of these to release an ATP. Because we have 3 conformations that we're cycling through. Okay. Other questions?
So like each one of these different like colors is doing the same thing.
They're all doing the same thing.
We're focusing on purple for this one, but they're all doing this.
They're all doing this, but they are offset from one another. Do you see how each of them is in, for each orientation of our gamma subunit here, each of these is in a different state of our 3 states. Okay. So let's look at some of the numbers we've learned previously and see if this makes sense. So previously we learned that if we have one NADH, right? You don't need to memorize this number because I'll give it to you and sometimes I'll change it. I'll make it a different universe. I'll make it a disease state or something. But one NADH pumped how many protons through the electron transport chain? 10.
Oh, 10.
Right. Let's go back and look at that figure.
It's 4 in the first one, and then it's
Yes. But if we put one NADH, make this a little bit smaller. All right, we put one NADH in the electron transport chain. By the time those electrons get to are accepted by oxygen, we have pumped 4, 4, and 2, or 10 protons, right? So that's what I'm talking about. So one NADH pumps 4H plus makes, how many ATP do we say usually you get from an NADH? 2.5 ATP. Okay, so we have 2.5 ATP is equal to 10H plus, right? But we just said that 3 ATP was equal to 9H plus. So ATP synthase. And then this is the electron ETC. The electron transport chain is pumping these protons so they can come back through, right? Do these numbers match up? No, they don't. Okay, that's the point, right? It's clear they don't match up. This is bigger, that's smaller, right? So what's going on? Where are the extra protons going? So these don't match. Where are the extra protons going? Let me add a sentence in here in the middle just to clarify. They don't match. All the protons aren't going to make ATP clearly, right?
Mm-hmm.
Did that answer your question, Lorenzo, or do you want to ask something else? Ah, okay. Do you know where they're going? It's not lost its heat energy, but that's a great guess, right? Okay. But where we're actually losing those, and that is a fantastic guess for that, is they're actually going to be used for secondary active transport. Okay. All right, so if we think about, we're making ATP in the matrix, right? If we look back at where we've seen this complex here, or the way I've been drawing them. So here, can you see I drew my little ATP synthase so that we were making our ATP inside the matrix? Right, I was doing that intentionally. And then every other time I've drawn the electron transport chain, there I didn't draw the ATP synthase with it. Let's go back. I'm going to give up if I don't find it quickly. Actually, let's go way back to the beginning. There. Okay. Also, well, I guess here, it's the opposite side to the intermembrane space, right? I wasn't labeling them at this point, but making it in the matrix. So we make ATP in the matrix, but where does ATP get used in the cell? Everywhere, including the cytosol. So we have to get ATP out of the mitochondria, right? So you have to get ATP out, must get out of the mitochondrial matrix. And we need to bring ADP and phosphate back into the mitochondrial matrix. I'll do ADP and do phosphate like that, so we don't have too many ands in the sentence. ADP phosphate must be brought back in. All right, so what we've got here, let me make this smaller so we can have this on the same page as this picture. And this is a great figure here. All right, what we've got going on, here's our ATP synthase drawn not very prettily, right? This is the C domain making ATP, all that sort of stuff, right? That's our ATP synthase. In the middle doesn't look like our more elaborate diagram, but we have to send out ATP and bring in ADP. Well, here we have a antiporter, right? Remember, an antiporter sends one thing across one direction, one thing across the other direction. That's going to let ATP out, okay? Now, ATP has a charge of -4 on it, and ADP has a charge of -3. You see any energy being used here? No. And so part of what's going on there is you may have a higher amount of ATP in the matrix, you may not. I don't actually know what the relative ATP concentrations are, but what do we know about, we've got our electron transport chain here, what do we know about the outside of the mitochondrial, well, the intermembrane space, I should say, for the mitochondria? We're pumping protons out actively using the electron transport chain, right? So it's positive out here. There's a lot going on in this figure. Beautiful figure. Okay. But we've got lots of positive charges out here, right? So what's going on for our net charge here for our ADP thing? That antiport.
Neutral.
Not neutral. We're sending out 4 negatives for every 3 negatives we bring in, and it's more positive outside. You see we're going down an electrical gradient there? So let's look at that. More negatives go out. Than come in. And it's positive outside. All right, so this is down an electrical gradient. All right, so the energy required to move that ATP out and bring the ADP in is all just provided by the gradient that's been created by the electron transport chain. And so no energy is really consumed there. Yes?
Okay, since you brought up proportions of hydrogens, I'm thinking it seems like there's not enough extra hydrogens to bring in a phosphate for ATP.
Right. So let's look. That's a good point. We make 2.5 ATP per 10 hydrogens, right? But we're not. So the ratios we're comparing, but we're comparing that to 3 ATP and 9. Does that make sense? So let's make. Let's do it like this. So if we have 2.5 gives me 10, how many would 3 give me? 30 divided by 2.5 is what? 15, don't know. I'm going to have to use my calculator. Embarrassingly enough. I'm hoping it's going to give me something like 12. Is what I'm hoping for. But 30 divided by 2.5. Look at that, it's 12. All is right with the world. Okay, so if I was to make 3 ATP, it would take 12 protons, right? And to make 3 ATP, I need to bring in, I haven't gotten down to where the phosphates are. I'm going to need to bring in 3 phosphates. Does that? Okay. Good question. Okay, so if we look here, so this we get for free. Not for free, we've made this gradient, right? But we're sort of degrading the gradient a little bit by putting some more negative charges out there. But here we need to bring a negative in, right? And it's more negative inside. All right, so to bring that negative in where it's more negative inside, we're going to need to go up an electrical gradient. And so this proton comes through. And there we're degrading the proton gradient a little bit as well. So this is up an electrical gradient. So a proton comes in, comes along to provide energy for transport. Okay, and that's an example of secondary active transport. All right, we directly consumed something. In this case, we took electrons from NADH, right? We made NADH to NAD plus so that we could pump protons across the membrane, and then those protons are being used to bring something across the back in across the membrane. Questions? And Jacob had the great question that we just established that we've now accounted for all the protons. Okay. With that, we are going to move on and talk about photosynthesis. So any questions about electron transport chain, all of that? And we talk about it, we break to talk about photosynthesis right now because there are things you'll see. There's lots of stuff in photosynthesis that's very similar to what's going on in our electron transport chain. Okay. And there's an ATP synthase and all that sort of stuff as well. All right, then we're going to talk about photosynthesis. All right, so photosynthesis is a simple picture, but essentially the point of this picture is to tell us that really most of the energy on Earth that drives life comes from the sun. You could say ecosystems. All right, so the sun is, sunlight's absorbed by photosynthetic organisms that make complex molecules, and then heterotrophs are going to non-photosynthetic organisms and photosynthetic organisms are going to break down those molecules for energy. Okay? And the reason why plants do this is not because they want to feed animals, but because they want to make it when the sun goes down, they need to make it till the next day. Okay? Or when the sun isn't down. Otherwise, if they were continually getting sunlight, they could just make ATP directly. And we'll see how that works. So thinking about our overall metabolism here, where we're at now is got photosynthesis on this diagram from your book, And really we're taking carbon dioxide and we're building it back up into 2 and 3 carbon intermediates. And that's really what we're going to do here. We're going to stop with that. But if I'm a plant and I have these pathways, right, and I build carbon dioxide up into a 2 or 3 carbon intermediate, what can I do if I need to make something more complex? So I could use the 2 or 3 carbon intermediate when it gets dark, maybe. But if I want to build something even more complex, like we know plants make starch, right?
Mm-hmm.
How would I get starch from here?
Like
But gluconeogenesis,
mm-hmm.
and then I would do glycogen, not glycogen synthesis, but starch synthesis, which we won't get into, okay? But we know pathways that can do these other things once we get to it'll be a 3 carbon intermediate, and it isn't pyruvate. Okay, so let's go ahead and talk about where this happens. We've got 2 different places in the chloroplast that happens. Here's a beautiful picture of a chloroplast, better than I could do. Okay, so chloroplasts, we've got an outer membrane, an inner membrane. We're going to ignore those. All right, and we're going to concentrate on these thylakoids. So we use thylakoids, which are these, is that how you say it?
Yeah.
I think I get corrected on that every year. But anyway, thylakoids. I'm sure your biology professors have been thylakoids is what they say.
Yeah.
Okay, they're probably right.
They also say niche, so they're
Oh, instead of niche?
yeah.
Niche just sounds better, but I've heard both.
Niche
They also
drives
say
me crazy.
cytokines or cytokines?
What?
Cytokines or cytokines?
It should be cytokines, but
Yeah.
anyway, I don't know for sure. And it's ligand.
Yes, correct.
We're
Like,
all right. So we've got this space that's outside the thylakoids. We call this the stroma, kind of like your cytosol of the chloroplast, and then we've got the insides of the thylakoids. And we've got 2 types of reactions or 2 sets of reactions we're going to concern ourselves with here. We have the light reactions, and these occur in the thylakoid membranes. And then we have what's called the dark reactions. And they occur in the stroma. Okay. And so if we were to zoom in on one of these, take one of these membranes and we were to zoom in on it, what it would look like is we'd have a membrane. Let's see, I want to get this right. This would be the stroma out here. This is going to be the thylakoid lumen.
That's inside the thylakoid.
Yeah, so lumen always means you're on the inside of whatever it is. You have the intestinal lumen.
Mm-hmm.
So thylakoid lumen, and so then what we have is, I'm going to draw this simpler here. We only really have one complex that's going to be pumping protons here. But what you can do is you can pump protons into the thylakoid lumen through a complex. That complex is going to, if we think about the whole photosynthetic light reaction complex, it's going to take water. So it's kind of the reverse of the electron transport chain, right? At the end of the electron transport chain, we make water. Here we're going to take water, we're going to make oxygen. which is why we have an oxygen atmosphere on Earth, okay, because of this reaction happening for over 1 billion years. When did that transition? Anybody know that? Remember? When did that transition happen? We went to oxygen. Yeah. Maybe, let's say 2 billion years, plus or minus 1 billion. Okay, so, but this is why we have oxygen on the planet. So we can pump protons in here. And then at the end of this, we're going to, if we're going to go straight through, we're going to make NADPH, right? Remember NADPH is used to make things? NAD, so we take NAD plus and we make NADPH, okay? That can be used to synthesize things in the stroma, which is where it's being made. So that's a good thing. And then we've got a proton gradient made by pumping these protons through, and then also you're going to be making things in the stroma. So that's the place that you want to let these protons come back through your ATPase. And you want to make ATP in here as well. Okay.
Where do the extra hydrogens go?
Where do the extra hydrogens go? Which extra
You
hydrogens?
have H2O.
Oh, I'm not counting hydrogens. Not sure. But they are all, science has accounted for all of them, I'm sure. I'm sure of that part. But we're not going to worry about it here. Okay. So we're going to get to the dark reactions. Let's see. Probably not today. We're going to get to the dark reactions, but what we've got is we're going to absorb light, and we're going to get this proton gradient going, so we can make ATP, so we can make NADPH, and these are going to be used to make our 3 carbon intermediates. Let's see, I want a new color. So these are used in the dark reactions, which is where we make our 3 carbon intermediates. The light reactions, their purpose is to make the NADPH and to make ATP. Question, we're going to go over the light reactions in detail now. Questions before I do that? All right. So in general, when people talk about well, actually, no, I'm not going to do that yet. I'm going to first talk about some other pieces of the chloroplast and about light energy, and then we'll get to that. So, okay, there we go. So the first thing I want to do is I want to remind you of some stuff from general chemistry. Remember that we kind of went over this last semester too, but remember that you can promote an electron directly from one level up to another, right? If we think simply about atoms, for molecular orbitals, it's more complex, and that's why we get these broad spectra. But if we were to just look at an atom, like we were to look at, let's look at hydrogen from the sun. Hydrogen has a lower energy, it has a lower molecular or atomic orbital number, and we have higher numbers, and then higher numbers, and then higher numbers. And if you take these, what this actually represents is an absorbance spectrum, which is cool. You could take these, and if you look at, say, this line, let's take the lowest energy line here, okay? We take the lowest energy line, that's going to be probably popping this up from here to there, okay? It might be there's enough going on in the sun that actually we've got electrons bouncing around. You get hit more than once. But essentially what's going on is you've got light, you've got these hydrogen atoms, you've got the light coming off the sun at the full spectrum, and then some of that light coming off gets absorbed by hydrogen and you get the electron popped up. Okay? And so if you were to look at the sun, you would see black lines actually. You can see black lines where that absorbance happens. It's an absorbance spectrum. Yes?
How did you say, you pointed at like one line and you said it was like the lowest energy. How did you determine that?
How did I determine that's the lowest energy?
Yeah.
I'm making an assumption without having, maybe I should read up about this more. and maybe you know something from physics, but I'm making an assumption that we don't have enough of the hydrogen in the sun that has electrons up here that our lowest energy line can be between one of these.
Okay.
So certainly if we're not in the sun, and we're doing chemistry in the lab, we always assume all our electrons are down here, and then we're either popping up to here, or there, or there.
Does that represent like energy levels you said?
Yes.
Okay.
So these are atomic, these are the orbitals, these are the rows of the periodic table we're looking at, okay? It's not an SPDF, it's first shell, second shell, third shell. But I might be incorrect. It might be that you have enough going on in the sun that you have some electrons populated enough here that you could actually get an absorbance from there to there. But otherwise, usually it's ground to something else. So we have this going on. Atoms can do this, and molecules can do this, right? They can absorb light. They can pop an electron up to a higher level, to an excited state, and then that electron can come back down. That electron could come back down and it could emit that light, re-emit that light. All right. And so this is what's going on in photosynthesis. You have light striking a molecule. It is going to absorb some energy that's going to become excited in terms of that, and then the electron can drop back down and we can give off that energy. All right. So we're going to see some of that. And we're also going to see electrons. The excited electron, you could pop up the electron, and you could see that electron just be taken away to reduce something. It's a higher energy electron. And that's an important thing that's going on here. Because remember, at the end of our electron transport chain, right, we had oxygen absorbed electrons, and it grabbed some protons, to become water, right? And we know water is super stable with those electrons. Why do we know that? Because water is stable. It's not reactive, right? It's a solvent. All of life happens. If it was reacting left and right with everything, it wouldn't make a great solvent, now would it? So we know water is stable. So if we're going to do the reverse of that, which is what photosynthesis starts with, we take electrons from water. We're taking something where they're really stable. We're taking electrons that are really stable when we're using them. And so that's hard. You need energy for that. All right. And so that energy is going to be captured. The main workhorse here is going to be chlorophyll, A. And it has a magnesium ion in it. We've got a porphyrin ring here like we saw for our hemoglobins. We've got a magnesium in there. And this complex is going to grab those electrons, and it's going to be used we're going to have those electrons, and we're going to use them to excite another protein. Okay? Now, chlorophyll A, if you look at it, okay, you guys, can you see that sword mostly? I've got a spectrum here, and over here I've lined up the colors of light from the sun. So this black line is the light we get from the sun. And if you remember from your lab's first semester, we did some chlorophyll isolation, right? And here's chlorophyll A. It's the purple line. So what problem do you see here? This is all the light we get from the sun and the intensity of it.
It only absorbs the lower sun.
Yeah, we're only absorbing here and here for chlorophyll. And actually, this is for photosynthesis. This is the part we use. Okay, this energy down here. And so we're missing all this other light that comes in. And so the point here is the sun gives off a broad range of wavelengths. Okay. However, chlorophyll only absorbs some narrow bands. So it's only going to absorb narrow bands of those wavelengths. And we can go ahead, even though we're not using this part, you can imagine the molecule absorb it, somehow give up some energy, and we come down to here, okay? But what you need is you want to absorb more than just those. And so what we have is light harvesting complexes. So chloroplasts contain light harvesting complexes that absorb all the wavelengths of light you could imagine. Okay, they absorb all wavelengths of light and then they transfer the energy to the chlorophyll A.
So all wavelengths go in, they just don't all get used.
They do all get used. A lot of the energy gets bled off. Okay, give me a second.
Okay.
But light harvesting complexes, and let me say, I said all, but let me say a broad range, a lot of, absorb a broad range of wavelengths, and then transfer the absorbed energy or part of the absorbed energy to the chlorophyll. All right, so what's going on here is we have a lot of these lower wavelength lights than the one we're going to use. What we're using here is 280 nanometers worth of energy, okay, right here. So if we use 280 nanometers worth of energy here, I'm sorry, not 280, 680. Okay, let me do this. It's, nanometers isn't a unit of energy, but you could convert it to energy, right, using Planck's constant once you've got the frequency. So we've got all these other molecules. What is higher in energy? 480 nanometer light or 680 nanometer light? 680. This is not higher in energy. This is lower in energy. This is red. This is blue.
Higher wavelength means lower energy.
Higher wavelength is lower energy. The longer the wavelength, the lower the energy. The longer the wavelength, the lower the frequency, the lower the energy. So these are higher energy, and what happens to happen is you have these steps where you end up with transfer of that energy from these higher wavelength energy packets to smaller ones. You're bleeding off energy as you come down until you get something that chlorophyll A will accept, which is that 680 nanometer wavelength packet of energy. Yes?
Is this one of the complexes? Like isn't there two, like a 720?
There is this, the complex is here, right? Or it's actually be transferred to it, but it's not, this is not part of that complex. This is absorbing the light to bring it in.
Is like the photo system? The photo system?
We have photosystem 1 and photosystem 2. We're not going to worry about that. That's not, photosystem, yes.
Sorry, I'm just confused where this was.
This is where we've got the energy that we got the protein P680.
Oh, okay.
Right?
Yeah.
This is we're going to grab, this is where we're going to provide, this is what's going to provide that with the energy.
Okay.
That.
Yeah.
I'm about to draw it out, so, but other quick questions? Okay, so then what this is it's a series, once we have this energy in chlorophyll A, we can transfer it okay? And what we get is we can transfer it and use it to actually do work. And we have the light reactions are a series of oxidation and reduction reactions. Okay, so what we've got is remember how I drew the interaction with water. So we have P680. Okay, is a protein. It's protein 680 nanometers of light is the wavelength of light it's going to absorb here, and it is it's oxidized. It's in its oxidized form. Actually, sorry, I need to move it down on the screen because I'm going to need space. I got 10 minutes. Okay. This is in its oxidized form. What I'm going to do is, if you've seen this in another class, I'm going to draw this different, and then I'm going to probably draw it the same as you've seen in another class. All right. There's a simpler, what you call a Z diagram, that doesn't really show the oxidation and reduction reactions. I'm going to draw it with the oxidation and reduction reactions first. It's going to be kind of a mess, and then we'll revert to the simpler Z form diagram, which I might have to draw next class. Okay, but I want you to get the feel for this right now. So we've got 680 is the most powerful biological oxidant. Known biological oxidant. Hopefully I didn't use space I need for my diagram. All right, so why do I call it that? Because what it has to do is it has to oxidize water. Remember, water's super stable. We have to oxidize water and make it into oxygen. All right, and that's going to give us P680 reduced. Okay, so we've taken electrons away from water here. What's then going to happen is this reduced form can get, so it's reduced, but to take the electrons away from water, it has to be more stable than water with the electrons, right? So it's less reactive than water. So that's not going to be any good for us. And so what we then need is it absorbs 680 nanometer light, and that causes us to get P680 excited. Okay, so we now have excited that electron or that molecule with the electron. Pops up there. And then what can happen is, I'm going to have to do this, I can then take that, now it's much higher in energy, and I can come in with what's called plastiquinone. It's like ubiquinone, okay, except it has a slightly different tail on it and slightly different functionality, but not for the business part of it. We'll look at the picture of it in a minute. And plastiquinone is going to be reduced to make plastiquinol. Okay, we've got that happening. And when that happens, so plastiquinone got reduced, so now we have to reoxidize our P680 at the same time. Okay, I'm not connecting the two circles like I normally do, because otherwise I won't be able to draw this at all. All right, then plastiquinol can dump off its electrons into a complex that's in the membrane. We'll just draw it in the membrane here. That's called cytochrome B6F. All right, and those electrons are going to move across this complex, right? And as they do, we're going to capture some of that energy, and we're going to pump those protons. We're going to pump some protons. Okay, and when we think about this, this is the thylakoid lumen, and this is the stroma. Okay, so we've got that going on. Oh, my stroma's in the way. And then on the other side of this, we're going to have plastocyanin is going to pick this up.
The deal or just the membrane?
It's just the membrane, right? All right, so we've got plastocyanin here. And plastocyanin, in its oxidized form, it has a copper in it. You get all these fun metals over here that we haven't gotten to see before, right? Just used to be iron. And plastocyanin is going to then become copper +1. So that's our reduced form. Yes. So it's like before we talked about cytochrome C and it's oxidized in a reduced form. And then it can dump off its electrons at to a protein that's called P700. So at this point, these electrons have the energy of 700 nanometer light. Or we're going to use, actually, that's what's going to excite this complex. So P700 not oxidized, I should say. That's going to be reduced. Okay. And then this can be excited by 700 nanometer light. To give us P700. And once again, now we're talking about excited, right? So we pump these electrons up in energy. We let them come back down. Then we pump them up again with light from the sun. That's where the energy is coming to drive this process. And then we're going to have these interact with something called ferredoxin. Which is an iron and sulfur containing protein. It is complex. It's not as simple as +3 to +2. Okay, so I'm just going to do oxidized and reduced here. Okay, and then finally, what's going to happen is ferredoxin reduced is going to interact with, and I don't want to extend my screen to the right here, because if I do, I know it messes up the posted notes. So I'm going to have NADP plus, okay, interacts here. It gets reduced to make, we'll bring this down a little bit farther, NADPH. And that's our final electron acceptor here for photosynthesis. It is NADP plus to become NADPH. And I didn't draw it here, but when ferredoxin gets reduced, our P700 gets reoxidized. Questions? And there's a simpler diagram for this, but it doesn't show the oxidation reduction pair. So I like to do this one first. And I'll do the other one next lecture. But are there questions? Okay. So next lecture, I'll probably also emphasize this, but sometimes, so what this does is this produces ATP and NADPH in exactly the ratios needed to make the 3 carbon intermediates in the dark reactions, okay? However, so you need a certain amount of NADPH and a certain amount of ATP. However, sometimes you want more ATP, okay? And when more ATP is desired, like I said, I'll do this again. You can take these reduced electrons from the ferredoxin, and they can come back and they can run through this complex again. So ferredoxin can come over, donate its electrons here, and we can run them through here. And so you could, without having to get new electrons, you could take these electrons and you can run in a circle here and not make the NADPH, but still make ATP because it pumps the protons across when that's useful.
That sounds like some good.
That's cyclic electron flow. Okay. I'll put it in and I'll label it in the next week. And then Sophia, for your this is photosystem 1 over here. This is photosystem 2 over here. Okay, you guys have a good hour. I'll see you soon.